What if you could listen in on the secret conversations happening inside a tumor? Or what if you could map out its entire hidden society? Well, today, we're going to dive into a really powerful computational technique that does just that, and it's completely changing how we understand and fight cancer. See, the first thing you've got to understand is that a tumor isn't just a simple lump of rogue cells. It's way more complicated than that. It's more like a bustling, chaotic neighborhood with all sorts of different residents. You know, a great way to think about a tumor is like a fruit smoothie. When you take a sip, you don't taste just one thing, you get a blend of everything, right? A tumor is exactly the same. It's this jumbled mix of the aggressive cancer cells, sure, but also the immune cells that are trying to fight back, and even the structural stromal cells that build the physical scaffolding for the whole thing. All of them are blended together, and that creates one single, really complex biological signal.
And that, right there, brings us to the huge challenge that has stumped cancer researchers for years. If you take a sample of that tumor, that smoothie, all you get is the combined signal, that one mixed flavor profile. So how on earth do you figure out the exact recipe? How do you tell how much of that signal is from the cancer versus the immune cells?
So to try and solve this, scientists have basically had two tools in their toolkits, and honestly, each one has some major trade-offs. It really comes down to this, seeing the blurry big picture versus seeing the super sharp details.
So, on one side, you've got something called bulk sequencing. Think of it like taking a blurry, wide-angle photo of a huge crowd. You can kind of get the general vibe, the average of everyone there, but you can't make out any individual faces. It's cost-effective, which is great, so you can study thousands of patients this way. But on the flip side, you have single-cell sequencing. Now, this is like a crystal-clear, high-definition portrait of just one person in that crowd. The detail is just, wow, it's incredible. But it's super expensive and technically really hard to do, so there's no way you can do it for everyone.
So you see the problem, right? We desperately need the best of both worlds. We need to get those crystal clear individual portraits, but we need them for the entire crowd. We need that fine-grained cellular detail, but from thousands and thousands of patients, because that's the only way we're going to find the real patterns that can tell us who will respond to which treatment.
And this is where our hero enters the story, a brilliant computational method that promises to do exactly that, to pull a crystal clear picture out of that blurry, messy signal. It's called transcriptomic deconvolution. Now, I know that sounds complicated, but stick with me. The transcriptome is just all the genetic messages flying around inside a cell. It tells you what that cell is actually doing right now. And deconvolution? Well, that's just a fancy word for unmixing. So you put it together, and it's basically a smart algorithm that can take our blurry photo of the crowd, that bulk tumor data, and digitally identify every single person in it, telling us not just who's there, but exactly how many.
Okay, so how does this magic actually work? Let's go back to our smoothie. First, you start with the mixed-up flavor profile of the final drink. That's your bulk tumor sample. Next, and this is the key part, you give the computer a reference library. It's like a flavor guide that says, this is what pure strawberry tastes like, this is pure banana, this is pure spinach, and so on. In the real world, that's a database of pure genetic signatures from known cell types. And finally, the deconvolution algorithm just does the math. It compares the mixed-up signal to all the pure ones and calculates the exact recipe, the precise proportion of each and every cell type in that original tumor.
All right, so we can unmix the data. Big deal, right? Well, yeah, it's a huge deal. It's an absolute game changer. Why? Because for the first time, this new, clearer picture lets us see things about cancer at a massive scale that were totally invisible to us before.
With this power, we can finally get a perfect head count of the whole tumor microenvironment, who's there and in what numbers. We can discover brand new subtypes of cancer that aren't defined just by the tumor cells, but by the entire cellular neighborhood they live in. This means we can start predicting treatment response, finding new biomarkers to track the disease, and even digitally pull out the pure genetic signal from the cancer cells themselves, stripping away all that background noise.
And look, this isn't just some theory stuck in a lab. This has been put to the test. We're talking about analyzing over 10,000 tumors, spanning 33 different kinds of cancer. And what did they find? They discovered you can basically sort all these different cancers into 6 distinct immune landscapes, 6 unique ecosystems that all behave differently. That is the kind of massive, large-scale insight that was just impossible before.
And get this, here's a totally mind-blowing example of what this can do. A recent study was looking at immunotherapy, trying to predict who it would work for.
And they found the most important signal, it wasn't coming from the cancer cells at all. Nope. Instead, it was the genetic activity in the structural stromal cells surrounding the tumor that best predicted whether a patient would respond. I mean, that completely changes where we even think to look for answers.
So where do we go from here? Well, if deconvolution is our amazing new digital microscope, the next big upgrade is basically adding GPS.
The next frontier is this incredible field called spatial transcriptomics. And this technology adds a whole new, absolutely crucial layer of information to everything we've talked about: location.
Okay, think of it this way. Deconvolution gave us the guest list for the party happening inside the tumor, right? We know who showed up and how many of them there are. But spatial transcriptomics, that gives us the floor plan of the party. It shows us where everyone is standing, and even more importantly, who they're talking to. And understanding those little conversations, those neighborhood interactions, that is absolutely everything when it comes to figuring out how a tumor grows, spreads, and fights back against treatment.
Ultimately, when you step back and look at it all, by digitally unmixing these incredibly complex biological signals, we're getting a view of cancer that's sharper and more detailed than we've ever had before. We are going from that blurry, averaged-out photo to a high-definition map. Which leaves us with one last, really powerful question. If we're getting this good at reading a tumor's secret recipe, how long will it be until we can start writing a brand new one?